Now come to the another example of the sex-linked inheritance or the X-linked inheritance in human, that is the hemophilia. Hemophilia or the hemophilic condition, this is the condition in which there is no proper clotting of blood, there is no proper formation of blood clot because of the absence of some of the blood clotting factor. In case of human, we have actually 12 number of blood clotting factors. Out of these 12 number of blood clotting factor, the blood clotting factor number 8, which is known as the anti-hemophilic factor, if the absence of anti-hemophilic factor 8, which causes the hemophilia A, or which is known as the classic hemophilia. If we have the lacking of the blood clotting factor 9, which is known as the Christmas factor, and which causes the hemophilia B or the Christmas disease. So these two, blood clotting factor 8 and the blood clotting factor 9, these are controlled by the genes which are present on X chromosome. It means that these are two linked genes, or these genes are the X-linked gene present on X chromosome. But another blood clotting factor 11, and which causes the absence of blood clotting factor 11, which causes hemophilia C, and such blood clotting factor which is known as the plasma thromboplastin. And such plasma thromboplastin, or the gene of such blood clotting factor 11, which is present on the autosome or on the autosomal chromosome. So here we have two types of X-linked hemophilia. One is the hemophilia A and the hemophilia B. So gene of hemophilia A and the hemophilia B, which present on X chromosome. And such hemophilia, this is the X-linked recessive disorder. Recessive disorder. It means that in case of female which contains or the hemophilic female which contains two X chromosome, it means that which contains two defective hemophilic alleles. If one hemophilic allele is present, then what happens? So if the female which contains one hemophilic or the defective alleles, then another X chromosome which contains the normal gene or the normal alleles and which compensate the defective hemophilic alleles. That's why the female will be normal but hemophilic carrier. So the hemophilic female, which is very rare in case of human, but in case of male, there is the one X chromosome and another Y chromosome. This is the recessive disorder. It means that if in case of male there is one X chromosome which contains one defective alleles, then what happens? Then there is the appearance of symptoms of hemophilia. So the man will be hemophilic. And there is no normal gene or the normal alleles on the Y chromosome. That's why there is no compensation of such hemophilic gene. In case of male, there is no carrier hemophilic, but in case of female, which will be the carrier but normal because of presence of one defective alleles, that's why the female become carrier. If one defective allele present in one X chromosome of male, then the male which will be hemophilic condition. So what is the actual hemophilia? The such hemophilic disease which was discovered by John Edward Otto in the year 1803, and the gene which is responsible for the formation of blood clotting factor 8 or the blood clotting factor 9, these are present on X chromosome, and such genes which is known as the anti-hemophilic globin gene or AHG. In case of hemophilic patient, because of minor injury, because of minor cut, which tends to bleed continuously and which will be fatal for the patient. And such disease actually found in case of royal family of Russia and the British. That's why such hemophilic disease also known as the royal disease. So X-linked gene present on X chromosome, and such X-linked gene is connected with the formation of blood clotting factor number 8 and the blood clotting factor number 9. And another hemophilia is there, and such hemophilia causes because of the blood clotting factor number 11 which is present on autosome. So this is the autosomal linked character. So now come to the how such hemophilic disease which transfer from one generation to another generation. And there is the big question is that in case of female, there is rare the presence of hemophilic disease because the hemophilic female which does not retain the maturity. That's why we have here 3 conditions. But we have seen that in case of color blindness, there are 4 conditions for the crossing. But here we will discuss about only three crosses. There is no hemophilic female. So let's start how it inherent from one generation to another generation. Now I am going to talk about the case number 1. So in the first case, I am going to cross about normal female with hemophilic male. Suppose that this is the hemophilic male. Suppose that this is the hemophilic male. So this is the parent generation. At the time of gamete formation, the female individual which will produce same type of egg, and the sperm which contains, or the male individual which contains 2 type of sperm, the 50% sperm contains X chromosome and other 50% sperm which contains Y chromosome. So these are the gametes. If the fusion taking place in between these 2 gametes, then the individual which will be female, and suppose that the fusion taking place in between these 2 gametes, then the individual which will be male. Suppose that this is the male which is the hemophilic. So this is the hemophilic gene which comes from the father to the daughter. This is the F1 generation. In F1 generation, there is the all daughter, they have the defective gene, so it will be normal, but the hemophilic carrier. The female will be normal but hemophilic carrier. So what about son? So all son, they become normal. If the marriage taking place in between hemophilic male with normal female, then in F1 generation, all females will be carrier and all male will be normal. Or otherwise you can say the 50% children will be normal and other 50% children will be carrier but normal. So this is the case 1. If the marriage taking place in between, carrier female with normal human, then what happened? Suppose that this is the parental generation. So this is the X chromosome which contains defective gene, and this is the 2 type of sperm. 50% sperm contains X chromosome, other 50% contains Y chromosome. Now female which also produce, these are the eggs, and the 50% egg having the normal X chromosome and other 50% egg having the defective hemophilic gene. If the fusion taking place in between these two gametes, then what happened? So the individual which will be normal, there is no defective gene. If the crossing over taking place in between these two, then here also you get the normal male individual. So this is the normal male individual. If the fusion taking place in between these two gametes, then the female individual which contains one defective gene here, hemophilic gene, that's why it will be normal, but this is the carrier, hemophilic carrier. It will be hemophilic carrier. If the fusion or the taking place in between these two gametes, then the male individual which contains the defective gene, that's why it becomes hemophilic. This is the male individual and this is the female individual. So out of two male individual, suppose that this is the male individual and this is the male individual. So out of two male individual, 50% male become normal and 50% male become hemophilic. In case of female, the 50% female will be normal. So this is the 50% female will be normal and other 50% female will be normal but carrier. So 50% female will be normal and other 50% female will be normal but hemophilic carrier. If the marriage taking place in between two carrier female with the normal male, then in F1 generation 50% female having normal and 50% female will be normal but carrier, and 50% male or the 50% son will be normal and other 50% son will be hemophilic. So this is the case 2. Now come to the case 3. So in case of another case, suppose that this is the case 3 and now I am going to cross in between hemophilic carrier female and the hemophilic male. Then what happened here? Suppose that these are the gametes and these are also gametes. Then what happened here? There is the defective gene, so it becomes carrier. If the fusion taking place here, then the individual which is male individual and this is the normal, there is no defective gene. If the fusion taking place in between these two eggs, then the female will be hemophilic, and if the cross taking place in between these two and cross or the fusion taking place here, then what happened? This is the defective gene and which will be hemophilic. So these are the gametes and this is the F1 generation. If the marriage taking place in between carrier female with hemophilic male, then in F1 generation the 50% son become the 50% son or male become normal and other 50% son or male become hemophilic. In case of female, 50% female or daughter become normal but carrier female, and other 50% daughter, daughter which will be hemophilic. hemophilic. This is the hemophilic female, and this is the hemophilic male. If the cross taking place in between carrier female with the hemophilic male, then in F1 generation we get 50% male normal and other 50% male will be hemophilic. And in case of female, 50% female become carrier and other 50% female will be hemophilic. But the hemophilic female does not retain mature condition, does not retain maturity. So this is the X-linked gene. But in case of Y chromosome, the very important gene is present, which is the Hollandric gene. And such Hollandric gene, which is the Y-linked gene, which transfer only from father to son, because the Y chromosome only transfer from father to son. That's why such Hollandric gene, which is responsible for development of hair on ear pinna, that's why which is known as the hypertrichosis. So the Hollandric gene or the Y-linked gene in human, which is responsible for development high hair on the ear pinna and which is known as the hypertrichosis. So this is the different condition of the inheritance of sex-linked gene in human. Thank you for watching.